students, I welcome you all for this video learning on the topic hormone receptors. Hormone receptors are large protein molecules that bind to specific hormones in target cells. Receptors are specialized cellular macromolecules or macromolecular assemblies that regulate cellular activity in response to neurotransmitters, hormones, drugs, or sensory stimuli. Thus, these receptors recognize a specific ligand, example neurotransmitter or a hormone, and they activate effectors within the cell. And the receptors are protein in nature, they are glycoproteins, and for a protein to be classified as a receptor, it must demonstrate the following characteristics like saturability, specificity, reversibility, reconstitution with function. Saturability refers to assuming a finite number of receptors for a cell, a dose response curve for binding of a ligand should be saturable, and it is expected to exhibit high affinity even though when the hormone is at its low concentration. And specificity. So, the receptors are expected to bind to its specific ligand, showing a high affinity and bringing out the specific biological response from the target cell. Reversibility. The ligand receptor interaction should be reversible. Reconstitution with function. The receptor should be functional in a reconstituted system. Classifying the receptors into two major classes, they can be classified as cell surface receptors and intracellular receptors. Under the category of cell surface receptor, it can be further classified into three various types, that is GPCR, G protein coupled receptor, enzyme associated or enzyme linked receptor, voltage gated ion channels. And under the category of intracellular, it could be classified into cytoplasmic receptor and nuclear receptor. After classifying, that is classifying the hormone into a broad category, they can also be classified based on the ligands that binds to the receptor. Under this classification, it could be a peptide hormone receptor or a steroid hormone receptor. Peptide hormone receptors are often transmembrane proteins. They are also called as G protein coupled receptors, sensory receptors, or ionotropic receptor. These receptors generally function via intracellular second messengers, which include cyclic AMP, inositol triphosphate, and calcium calmodulin system. And steroid hormone receptors are generally soluble proteins that function through gene activation. Their response elements are DNA sequences, which we call it as promoters, that are bound by the complex of a steroid bound to its receptor. The receptor themselves are zinc finger proteins, and these receptors include those for glucocorticoids, estrogens, androgens, thyroid hormones, calcitriol, and the retinoids, that is vitamin A. Based on the location, the receptors can be classified as cell surface receptors and intracellular receptors. Lipophilic hormones, that is lipid soluble hormones like thyroid and steroids, can pass through the nuclear and the cell membrane, and these hormones target the specific DNA sequences by diffusing into the cell. And these lipophilic hormone receptors, upon binding with these ligands, they undergo conformational changes and influence the transcription process. Whereas hydrophilic hormones, which are the water soluble hormones, are lipophobic in nature, and thus these ligands cannot diffuse through the cell membrane. Example includes catecholamines, glycoproteins, insulin, etc. These receptors for these hormones are located on the surface of the plasma membrane. Thus, the hormone receptors are found in the target cells, cell membrane, cytoplasm, or nucleus. So this is the pictorial representation of the cell surface receptor and the intracellular receptor. In the cell surface receptor, we can see the receptor is a transmembrane protein which is present on the plasma membrane, and the ligand binds to the surface of the cell surface receptor, and once the ligand receptor complex, then it leads to the intracellular response. And in the intracellular receptor, the signaling molecule, that is the hormone, which is lipophilic in nature, easily passes through the plasma membrane, enters into the cytoplasm, and it binds to the receptor which is present in the cytoplasm, or it can also bind to the receptor which is present in the nucleus. Based on the mechanism of action, the receptors can be differentiated as fixed membrane receptors and mobile receptors. Now, looking into the mechanism of action, in fixed membrane receptor, the hormones that are proteins or amines in composition, such as growth hormone, ADH, oxytocin, insulin, adrenaline, FSH, TSH, etc. shows this mechanism of action. That is, these hormones are water soluble and cannot pass through the lipid membrane, and they have their target receptor on the cell surface. Hence, the receptor are fixed on the cell membrane, so hormone can bind on the specific receptor. Binding of hormone on specific receptor on the target cell activates the enzyme adenylate cyclase in the cell membrane and causes the production of cyclic AMP, which is a second messenger. And this cyclic AMP acts as a second messenger, and it diffuses through the cell membrane, and it activates various protein kinases and enzymatic reaction to cause biochemical changes. After the target cell responded to the changes, CAMP is deactivated by a group of enzyme phosphodiesterases. Coming to the mobile receptor mechanism, the lipid-soluble hormones such as steroid hormones and fatty acid hormones can easily pass through the plasma membrane, and they have the receptor inside the cell floating in the cytoplasm. Binding of the hormone to the specific receptor activates the enzymatic activity of the cell for biochemical changes. Some hormones like testosterone, progesterone, estrogen, cortisol, and thyroxine have their receptors localized inside the nucleus. The hormone receptor complex are carried inside the nucleus. The hormone receptor complex, it initiates transcription of the DNA to form specific mRNA, and the mRNA initiate protein synthesis in the cytoplasm. The protein causes biochemical changes in the cell. Thank you, students.